Hello, everyone. We're giving the audience a few seconds to enter from the waiting room. Thank you for your patience. So today we have a large group of presenters, really honored to be here with so many colleagues today. This is Sabrina Paganoni, and I'm connecting from the HEALEY Center for ALS and Mass General. And today I'm joined, we have a great group. I'm so excited to have our colleagues from Denali. And I'm going to ask everyone to briefly introduce themselves so that you'll know who is on the call today. And I'm also here with some colleagues from the HEALEY Center. So I'm just going around the Zoom squares I see here. I'm going to start with you, Danna.
Hi there. Yes, good afternoon, good evening. I'm Danna Jennings from Denali Therapeutics and the clinical lead for this DNL 343 program.
Thank you. Sydney?
Hi there. Pleasure to join you all on this webinar today. I'm Sydney Gardner, and I am the patient advocacy lead at Denali for the ALS program.
Deepika?
Hello, everybody. Deepika Tamoli, and I'm the clinical operations lead for the DNL 343 ALS program at Denali.
Thank you. And from the Healy Center, as you know, every week I'm here with Catherine and Mimi. So I'm just going to ask Catherine to introduce herself before Mimi.
Yeah, sure. I'm Catherine Small. I'm the patient navigator for the HEALEY platform trial.
Great. Mimi?
Hi everyone, I'm Mimi. I'm one of the admin assistants for NCRI.
Thank you so much, and thank you for setting up these webinars, which I think are a great forum for the community. So tonight, I know that many of you remember my colleagues, Dr. Lori Chibnik and Suma Babu. I know maybe you can introduce yourselves. You have been frequent guests on this webinar series, so thank you again for being here. Lori?
Sure. Hi, I'm Lori Chibnik. I'm the biostatistician. I've been approached and asked if I was the biostatistician, so that is who I am.
Thank you. And Suma?
Hi, everyone. I'm Suma Babu, ALS neurologist at the HEALEY Center and co-lead for this particular regimen with Dr. Paganoni.
Wonderful. Thank you so much again. Always a pleasure to be here. And tonight, really an honor to turn this over right away to our colleagues from Denali. Danna, take it away.
Great, thank you so much. And today I'm planning to share the top line results from the DNL 343 Regimen G of the HEALEY platform trial. And for those that are not aware, I just want to say up front that the primary and the secondary endpoints for Regimen G were not met. Of course, this is terribly disappointing, a disappointing result for the ALS community, but especially, for the study participants and other individuals and families living with ALS. I do want to just take the time to thank the HEALEY team for this opportunity to share these results. And really the reason to share negative results is to try to learn as much as possible from the trial and continue to advance more effective therapies for ALS. So let's go to the first slide. So next slide, please. Yes. So let me start by just saying that at Denali, we feel incredibly fortunate to have had the opportunity to join the HEALEY platform. We developed just wonderful relationships with the HEALEY leadership, the site investigators and coordinators, and really the community during the course of this study. So here you can see that the pictures of the HEALEY leadership are shown here. Merritt Kudkowitz, the platform trial PI, and then Suma Babu and Sabrina Paganoni, who were both on the call, were the Regimen G lead investigators. And the HEALEY platform really offered several advantages, some of which I think are listed here on this slide. But there's a very streamlined approach to the startup process, and this is really given that the sites are really already in place, and it utilizes a central IRB. But there's also the very efficient and rapid enrollment. And honestly, this exceeded our expectations in terms of enrollment for this study, which was really a fantastic benefit. But I'd say most importantly, or one of the really important pieces of the platform design is that it supported the efficient collection of the data and also the analysis of data that could ultimately be decisional. So that I think was really a huge benefit from a sponsor and industry partner perspective. So how DNL 343 entered the program? DNL 343 entered as Regimen G at the very bottom of this list. And Regimen G was running concurrently with Regimen F and shared the same placebos. So Regimen G and Regimen F placebos were used for the primary analysis. Let's move forward to the next slide. Right, so I just want to start by talking a little bit about the therapeutic hypothesis, and I thought it would be helpful to provide a high-level overview of the proposed mechanism of action for DNL 343 in ALS. So DNL 343 is a drug that activates EIF2B. And really the overarching hypothesis for this mechanism of action of DNL 343 is that it protects against the integrated stress response, or the persistently active integrated stress response. Sometimes we call the integrated stress response ISR for short. So you'll see that in the slides. So the ISR is sort of a change or adaptation that occurs in response to some sort of stressful event, including, for instance, like an ALS risk gene. Now, the integrated stress response triggers inactivation, turns off the EIF2B, causing interruption of normal protein production and the formation of stress granules and the formation of abnormal TDP-43 clusters. And TDP-43 clusters, as you may be aware, or pathology, TDP pathology is present basically in all individuals with ALS. So this process of the integrated stress response just ultimately results in neurodegeneration. And if you just move forward the slide, so the proposed mechanism of action of DNL 343 is that it is an EIF2B activator. So it actually turns back on that EIF2B protein complex. And this has the potential to normalize protein production, reduce the stress granules and the TDP-43 clusters, and then ultimately to restore normal neuronal function. So let's move to the next slide. So for DNL 343, we have conducted 2 studies in ALS participants, which are shown here. So the first study was a phase 1B study in 29 participants that were randomized for a 28-day double-blind treatment period, followed by an 18-month open-label extension. Now, I'm not going to go through the results of this study, but I want to just say that the endpoints or the main goals of this study were to evaluate safety, to evaluate the exposure of DNL 343, the DNL 343 level in the blood, the biomarkers of neurodegeneration, including NFL and ISR pathway biomarkers. And just as a top-line result from this study, we were able to demonstrate robust reduction of ATF4 protein and CHAC1 gene expression, which are these ISR pathway biomarkers, demonstrating that we were actually hitting the target that we were intending to impact, and that occurred at doses that were safe and well tolerated. So ultimately, the data from this phase 1B study supported moving into the HEALEY platform Regimen G. And in that study, and that'll be really the focus of the discussion here today, we randomized 249 participants with ALS. and they were randomized for a 24-week double-blind treatment period and a 52-week active treatment extension where everyone received the drug. The main goals of the HEALEY Regimen G were to demonstrate clinical efficacy, evaluate biomarkers of neurodegeneration, including NFL, evaluate pathway biomarker, ISR pathway biomarkers, as well as safety. Next slide, please. So in just a little more detail around the study design, I'm going to start on the right-hand side of this slide with the key inclusion criteria, which you can see included participants that had less than or equal to 36 months since their symptom onset, a vital capacity of greater than or equal to 50% predicted. And then for those that were on a standard of care, they were required to be on a stable dose. So individuals that met this key inclusion criteria, there were 249 participants that were randomized to DNL 343 or placebo. So you can see that 186 total participants were randomized to DNL 343 and underwent the 24-week double-blind period, and 144 went on to the active treatment extension. I will say that the active treatment extension is now in the process of closing out as a result of the lack of efficacy. And in terms of placebo, we had 63 participants that were randomized to Regimen G placebo, and of those, 54 went on to the active treatment extension. And in terms of shared placebo, there was 139 participants that were on the shared placebo being Regimen G plus Regimen F placebos. So with respect to the top-line results, here I'm going to focus on the primary and secondary endpoints, and these really encompass the top-line results. So the primary endpoint was the change from baseline in the disease severity, which was measured by the ALS FRSR total score and survival. And this used a model which is called the Bayesian approach or the Bayesian model. And the secondary endpoints were measured at 24 weeks at the end of the double-blind period. And here we measure the change from baseline in ALS FRSR, the CAFs, SVC or vital capacity percent predicted, serum NFL, muscle strength based on the handheld dynamometry, and then also survival. Now, in terms of exploratory endpoints, this is really beyond the scope of what I'm going to talk about today. But nonetheless, there are a number of biomarkers that are still undergoing, biomarkers and also patient-reported outcomes that are undergoing analysis. Next slide. So this is the demographics and baseline characteristics. This is a very busy slide, so I'll just talk about a couple of the differences that are highlighted here. So the way we look at demographics is evaluating the demographics for each particular treatment group. So the first one is DNL 343, 200 milligrams QD. And then the second column here is the shared placebo, so the Regimen F and Regimen G placebo. And then the final column is the Regimen G placebo alone. So I would say largely the groups were very similar. A couple things are highlighted here in bold, which I'll just point out that there were a higher percentage of females in the DNL 343 treatment arm. There was also an increase in the percentage of individuals that had a bulbar onset for their ALS. And then in terms of baseline serum NFL, the DNL 343 group also had a little bit higher NFL at baseline. Let's move to the next slide. So in terms of the participant disposition, This, I would say, is a very well-conducted study, and we had a high rate of retention in this trial. So starting on the right-hand side, you can see in the orange box, the DNL 343 Regimen G enrolled 186 participants. 84 completed the double-blind period, 84%, sorry, completed the double-blind period, and 77% went on to the ATE. And in terms of the shared placebo, the black box on the left-hand side, there was 139 that were enrolled in the double blind, and 91% of those participants completed the double blind. And the gray box, the Regimen G placebo, and enrolled 63 in the double blind, and 87% of those participants completed the double blind, 86% were entered into the ATE. So a very high rate of retention. Next slide, please. So now we're going to move into the results of the study. So let's move directly into the primary endpoint. So the primary endpoint is shown here, and it again, as I mentioned previously, utilized the Bayesian model to evaluate the change in ALS, FRS-R, and survival in the 24-week double-blind period. So you can see that we were comparing here the DNL 343 participants, which are shown in the orange dots and the orange slope line. And we're comparing those to the shared placebo, which is in the triangle, the black triangle, and the gray slope line. And those lines basically overlap each other. So they're very much overlapping. And this over this 24-week period. So this shows that DNL 343 really showed no effect on progression as measured by the ALS FRS-R scale. Next slide. And in terms of secondary endpoints, we also looked at the ALS FRS in a different way, just looking at the change from baseline in the ALS FRS-R. Again, at 24 weeks in the double-blind period. And very similarly, we compared the DNL 343, which is in orange, to the shared placebo, which is in gray here. And there was a similar rate of progression on ALS FRS-R in DNL 343 treated in the shared placebo. And the rate of progression is, pretty similar to what's noted in the literature to be pretty standard for ALS, which is approximately 1 point per month. Next slide. We also measured and evaluated muscle strength. And here we used the mean percent change in handheld dynamometry of both the upper and lower extremities in the 24-week double-blind period. And you can see on the left-hand side is, again, the mean percent change in the upper extremity. And DNL 343 is in the orange compared to the shared placebo in gray. So there was a little bit smaller percent greater worsening, a small percent greater worsening in the shared placebo in the upper extremity. However, that was not replicated in the lower extremity, which you can see that the lines are more overlapping. So really no meaningful difference in handheld dynamometry or muscle strength. Next slide, please. So next we're going to talk a little bit about the top line results for the biomarkers. So let's move into the next slide, which really just describes our strategy at the beginning of the study in terms of biomarkers. So one of the things we were thinking about as we designed the study was demonstrating both pathway engagement, does DNL 343 inhibit the ISR, the integrated stress response, but the focus for the top line results was really the NFL the NFL levels and measuring whether DNL 343 had any impact on NFL both in the serum but also in the CSF. So the biomarker strategy focused on NFL. We do have some other exploratory biomarkers, which again are beyond the scope of what I'm going to present today. They're still undergoing analysis, but we'll definitely be able to present those at a later time. So next slide. We're going to move directly into the NFL results. So here on this slide, we evaluated both the serum and CSF log transformed NFL. And if you look at the graph on the left-hand side of the screen, you can see that this is the serum NFL. And we looked at this both in the double-blind period, which is up to 24 weeks. And here we compared the DNL 343 to the shared placebo. And there's really no difference at 24 weeks from baseline. And then ultimately looked at this moving all the way out to 52 weeks, with the idea that maybe 24 weeks was not long enough, but you can see that again, here we compared it to the Regimen G placebo because that was the data that we had, but ultimately there was no difference or no reduction in serum or CSF or serum NFL at 24 weeks or 52 weeks. On the right-hand side of the screen is the mean log transformed NFL in the CSF. So we looked at the CSF NFL, and certainly those lines were very much overlapping when compared to the Regimen G placebo. So no difference and no reduction in serum or CSF NFL with DNL 343 treatment. Next slide. So this slide just describes the safety endpoints for the study, and this is an overview of the safety and tolerability for the 24-week double-blind period. Again, not going to go through every number on this slide, but just to say that in the DNL 343 treatment arm, there was a higher percentage, a modestly higher percentage of individuals that reported any treatment emergent adverse event. DNL 343 treatment group also had 3 individuals that had treatment-related serious adverse events, and those included a transaminase increase or an increase in liver function tests, an ECG change that was abnormal, and then also drug eruption or a rash in one individual. All of these serious adverse events were non-fatal. In terms of withdrawal, interruption, and reduction of study drug, there was an increase in these events and TEAEs that were leading to withdrawal, interruption, or a reduction in the DNL 343 treated group. So the most common treatment emergent adverse events in this study that were more common in DNL 343 treated versus placebo treated participants included fall, fatigue, and urinary tract infection. We also noticed that in the DNL 343 treated group, there was an increase in transaminase levels, and that is, again, the liver function test in 10% of DNL 343 treated patients compared to 3% of the placebos. With that, there was 1 withdrawal and 6 with dose interruptions, but 4 of those were successfully re-challenged with the medication. Next slide. So just to summarize, the results of the HEALEY platform trial, Regimen G. Again, the primary and secondary endpoints of Regimen G were not met. And the HEALEY platform trial, I will still say it was a very well conducted study. and importantly, I think provided us with decisional data that we can that we can move forward on. So, given the lack of treatment effects that were observed in this study, further investigations of DNL343 in ALS are no longer warranted. And data from this study, the Regimen G DNL343, will continue to be evaluated to help inform future trials in ALS. Next slide, please. And most importantly, we are just incredibly grateful for all of the study participants and their families, investigators and site staff and the HEALEY leadership for their generous contributions to the DNL343 clinical development program. I mean, we really honestly could not have done this study without the help of this entire research community. So thank you very much.
Thank you so much, Danna. I want to echo the gratitude to the ALS community on behalf of the Healey Center team as well. It really takes a lot for participants to participate in these trials, and time and effort. And we're very grateful for everything that every participant has done to participate in this trial. Thank you so much. Danna, thank you for your presentation. I think we have time for maybe one question. There's one question that has come in terms of when will maybe this is a question for Sabrina, when will the RGG participants find out about their treatment group assignment?
Yeah, absolutely. We are committed to sharing the information with the participants. We've done that actually in previously closed regimens. And so we are working now on closing the database and collecting all the data from this regimen. And as soon as the process is completed, which I believe should happen over the next few weeks, we will be able to release the treatment assignment groups. I do want to mention that at the Healey Center or the company or at the coordination center, we do not know the identities of the participants. So this type of communication will always go through your site. So in other words, we want to make ourselves again available through webinars and conferences so that people can reach out and ask questions. But in terms of individual specific information, such as treatment assignment, that will be provided by your study team. So your doctor, your nurses, the people that you normally see at the site.
Thank you, Sabrina. I think with that, we come to the close of this webinar, and thank you all so much for attending this webinar. We appreciate all your time, and thank you, Denali team, for an excellent presentation.
Thank you so much. Let me just add that, it's really important that we also have these conversations and share the data, in a timely manner. We will be working on publications. We're gonna, present these upcoming conferences. I think we all want to make sure that we share all the data and the learnings, because while there's no new drug based on this regimen, all the learnings and all the data and samples that have been collected will continue to contribute to ALS research. So I do want to end on a positive note in terms of the contributions of all of you that participated in the trial. Actually, that those are definitely not lost. We're going to continue to use all the data and samples for additional ALS research, and that's also very important. So just wanted to say that. And I don't know, Danna, if you want to give us the final word.
Yeah, no, thank you very much. And again, many, many thanks to this community for, allowing us the opportunity to work with you.
Thank you so much.